So our second half of the program is a second example of a Frontier Research Program grant. As I said earlier, one of our very earliest ones, which I think, Linda, you were the PI of. I'm not sure. Maybe both of you guys were the PIs. And it was an idea that was in a different realm of cancer research, trying to develop molecules and technologies to allow doctors to do a better job in the context of surgery to remove cancers more effectively. And you'll hear in detail how it came to be and where it stands. And it's another example where we went from a concept to early stage funded research through this program in the laboratory, now to a commercial entity, Lumisell, which is advanced into the clinic to the benefit of patients. And I can tell you from the perspective of somebody who oversees research in an institution such as this, there is nothing more gratifying than we see examples of things that we funded, sometimes the high-risk ones, coming to fruition in such a way. So I'm really pleased and really proud of the accomplishments of this next group. The first speaker will be David Lee, whom I've known for a very long time. We were neighbors, actually. David was an MIT undergrad and got his master's degree here. Went off to a distinguished career at Analog Devices, and then made a shift, using his engineering background to think about cancer in new ways. Actually became highly connected to the Koch, worked in the Koch for a period of time, and then became involved in a series of projects, actually, that we were involved with. We founded T2 Biosystems together, and he then got engaged with these folks to do Lumisell, and that's really become a full-time occupation. He's the chief scientific officer today, formerly the chief executive officer of Lumisell. He's going to be joined tonight by two of my faculty colleagues at MIT, Linda Griffith, who's a faculty member in the Department of Biological Engineering and a real pioneer in understanding the cell biology of cancer and other diseases, and Moungi Bawendi from the Department of Chemistry, and he's an expert in an area of nanotechnology we call nanocrystals, and specifically quantum dots, and the use of those materials for various applications, actually. In this context, it's biology and medicine, but he's used them in many different applications, from electronics and solar energy and so forth. So again, it's a real fusion of talent, which we highlight here at the Koch Institute, brought together, hopefully, for improved outcomes for cancer patients. So we'll start with David.
Well, thank you, Tyler. You actually took away a little bit of what I was going to talk about, so I'm going to have to rethink. But anyhow, Linda, Moungi, and I are really pleased to be presenting to you something that we developed. What we have done is we focused on a huge unmet need in cancer surgery. You might not know it, but nearly 30% of all breast cancer surgeries have to be repeated because the surgeon can't find the small microscopic residual cancer left behind. And that's true in many other solid cancers, not just breast cancer. The challenge is not finding the tumor. That can be done with X-ray and palpation. The challenge is finding the microscopic residual cancer that's between the tumor and the healthy tissue. What hospitals have attempted to do in the past is adapt or adopt conventional technology. Here you see a $20 million adaptation of MRI for cancer surgery. The difficulty here is MRI can't find microscopic tumor. Anything below a millimeter is a challenge, and that's really where all the residual cancer is. And also, it doesn't work very fast. It's hard to use in a OR setting. So what we did was developed a sophisticated and exquisitely sensitive imager. And the way it's used is the tumor is removed by the surgeon. The handheld device is used in the cavity. It finds the tumor, lights up red on the monitor. They go back and take out the tumor that was left behind. Then they re-examine the cavity. This all takes place in about the amount of time that you see there, a few seconds. The novelty of this approach came from the recognition that pixels of a camera are smaller than tumor cell sizes, and so we should be able to image, find, and detect single cells of cancer, but in a wide field of view, which is what's needed when you're doing surgery. What we detect, though, is fluorescence. The fluorescence comes from our imaging agent we call Lumicell 15. Nobody knows why it's called Lumicell 15. And, well, somebody does. And the novelty of the imaging agent is that it doesn't target the tumor because, as you saw, the tumor is removed and sitting on the table. What it targets are the cells that are in the margin, a mix of tumor cells and immune cells. To date, what we have accomplished is we've finished our phase 2 clinical trials in breast, started others in GI, and have been very successful in the breast cancer, expect to be as successful in the others. We've started phase 3, which means that we're about a year away from from FDA approval, going to market. And we did this all with a single platform, which will also address 5 other cancers besides breast cancer that we know of. And each of these is a market that's about $150 million a year, addressable market. And with 6 cancers, that means that we're looking at about $1 billion a year addressable market. So we did this whole thing on $15 million of capital, and then some additional NCI funding. That was my intent from the beginning, to demonstrate that we can do this with low capital. We think that the next 10 cancers will be developed with less than $10 million. Sorry, the next 5. Pharma typically spends between $100 and $150 to $200 million on the development of a new molecular entity, a new imaging agent to take it all the way through clinical trials. And so you might say, okay, how'd you do this? As Tyler said, this was all about the team. And it all started, I'll take you through the journey. It started more specifically in the driveway between my house and Tyler's house. If you remember clearly, I cornered you. And this was shortly after my wife died of breast cancer. I decided I didn't want to work on semiconductors and accelerometers for the Wii, which some of you might have. I was responsible, and for the motion detection. And I asked Tyler if there could be something I could do in cancer, and he opened the door for me to come to some meetings. I'm an inventor, and I thought of a new approach, a new diagnostic, which became a project then, as Tyler said, became a company, T2 Biosystems. It was very exciting, but it wasn't going to be cancer. So I went back and met with Tyler in his office this time, and he pointed me towards tumor margins. So I went off and thought about that and came up with a concept of how to do it, which interested Moungi and Linda. And at that point, we decided to submit a proposal under the Frontier Research Program, and we're lucky to be funded by Kathy and Gary Marple and their fund. Gary's right here. And as you've heard before, this funding is critical, and particularly for a technology which at this time was all device. It's very hard to get funding for device technology, especially when you haven't built it and you have no data. And so in the academic setting, you typically want to have an R01 funded grant. And the chicken and the egg problem is you got to have preliminary data, but if your device is what you need for the preliminary data, you get nowheres. And so as soon as we built the device, demonstrated that it worked, at that point we formed a company, had funding, And Moungi, Linda, and I then set about to create a world-class team. And this was really important. It's from my years of experience. The prior years before Analog Devices, I was at Arthur D. Little, managed the technology business. And I knew that we needed a diverse team to handle the future problems. MIT, Duke, and MGH, plus the Lumisell folks. And the core of our team was always data. We used data, we developed simulations, we predicted the performance of the device and the drug, and we used that throughout our development process to accelerate it. And it had some unexpected benefits. Halfway through our phase 2 trial, the FDA put us on clinical hold. Anybody that's been in a trial knows what kind of trauma that creates, especially with your board of directors. And they wanted us to run a separate trial at different doses. We had a simulation, we had data and our model. We thought we knew what would happen. We put that in front of the FDA. They're a bunch of scientists. They accepted our model, took us off clinical hold, and we were back running. And that vindicated, I believe, the approach. We stuck with data and models throughout the development. And that got us in through phase 2 and now into phase 3. There were a few other bumps on the way. So early on, as you could tell, it was a device company, and that was the center of it. And we hit what I like to call a wait what. We were about to license the imaging agent from a company, and as we poured through it, we realized they really hadn't developed it very far, didn't have animal data, and we couldn't justify going down that path. So we became a device and a drug company. And another wait what was when we met with our surgeons and we found that they didn't want data, they didn't want to look at an image, they wanted a yes, no, and where is the tumor. So that meant that we had to become a device, a drug, and a decision software company. And that is who we are. This is the team that did that. There's 8 people there, 7 of whom met every week since 2008. And that is really important, and especially when you consider all the skills that we needed. They're all tallied up here. This allowed us to address all the problems that came up and all the opportunities, allowed us to become a drug development and a software development company. And behind this was our culture. As I said before, data was the boss. We had no dominant voices in the meetings except for the data. And we questioned everything and looked for the data to support it. We made what I'd call lean choices, always looking for the cost at the end of this. We had in mind that we were going to be a low-cost solution one way or another right from day one. And that comes from my history in commercial world. And we kept the cycle as a 7-day cycle, and that was really critical. I can't say enough about that. Having senior professors willing to put in the time every week for 9 years is amazing, and it keeps the momentum of the development. It keeps the share of mind with everybody. You don't forget what the issue is because it's only 7 days before when the issue came up. And the cycle was to identify a problem, think about the data we needed, design a benchtop or an animal experiment that would get the data and get that within approximately 7 days. Some of these took longer, but that was the pace, and that was how we ran the business. The thing about this is that good ideas also could be explored because we had a diverse team, so that when there was a new off-the-wall idea, there was always somebody in the group that had some experience in that area and could tell us whether it was feasible or reasonable to explore it. The converse is that we could take in any idea, and if it was a bad idea, it only lived for about a week. And so that kept our costs manageable when we wanted to explore unusual things. We saw things in our data that suggested really quite unusual ways to alter the contrast. We were able to explore that quickly and discard it because we had this rapid cycle. And so with that, I'd like to turn this over to Linda Griffith. She'll talk about the drug development. You got it.
Thank you so much. Let me turn this on. So it's always a somewhat Rashomon-like experience to talk with your colleagues about an experience that happened so many years ago and how did it start, what really happened back then. And my memory is a little clouded because soon after we started our project together, I was diagnosed with breast cancer, and actually a fairly aggressive kind. So I've had chemotherapy wipe out a little of my brain cells, so I might give a little bit different version than David or Moungi would give. But at the time that David came to MIT, he is an incredibly open-minded person. And he had this idea with Tyler to look at tumor margins, influenced partly by his own personal experience with breast cancer. And he went around MIT on a listening tour, talking to people, just listening and really trying to learn what they were interested in. And he came to see me because somebody said maybe Tyler should come see me. And at the time, I had a research grant that's a special kind at NIH called a Transformative R01, in which we were thinking about communication networks involved in tumor invasion and all kind of inflammatory disease invasion in which numerous processes, proteases, activation of growth factors, et cetera. And so he came to see me at a time when we were thinking about certain kinds of biological processes that actually happen at tumor margins. And he wanted to look at margins. So we had a really good conversation. And at the same time, I was teaching physical chemistry together with this amazing physical chemist, Moungi Bawendi. And we had a great teaching relationship. And this is one of the things that really is the secret sauce of MIT teaching. And so this idea to work on the tumor margins and think about how we could detect cells seemed fantastic. To work with David, to work with Moungi, what could be more exciting, more fun? That team is amazing. And one of Tyler's former postdocs, David Koch, also. And so, and David prepared these slides, which are really the history, which we all do agree on. The conventional thinking, meaning what David had heard on the street up until then and brings into our group, perhaps, was to target the tumor itself. And there are many antigens on tumor cells for which there are antibodies, or you could maybe make a molecule like Dane Wittrup does here in the Koch that is evolved to recognize the antigen. So an antibody, Herceptin. Who knows what Herceptin is? All right, so some people know breast cancer patients or work in the field. It's an antibody that targets an antigen overexpressed on many kinds of tumor cells, particularly in breast cancer. First big blockbuster biotech tumor drug. And it targets something called HER2. And I studied HER2, so I knew a lot about this. And actually, I ended up on the front page of the New York Times because when I got breast cancer, I was ambiguously positive. And there was a big drama about how to do the testing and what to do with patients, and I had to make a decision about my treatment. So I knew a lot about the conventional thinking where you target things that are unique to tumor cells. But we also were very aware in the world I live in, which is in biological engineering, where many people study cancer from systems biology, and it seemed reasonable to think more about broader processes that happen at the margins of anything where there's a lot of active invasion and so on that involve the immune cells that are in the tumor margins. So we got the idea together to target this whole activity in the margin, which is not only the tumor cells, which may all be very, very specifically different, but actually general activity involved within inflammation and tumors that would occur at the margins. And so this was a bit outside some of the conventional thinking. There are many, many kinds of tumor imaging agents that's targeted specifically at antigens. And so when we first showed our work and published the very first in-human trials of a protease-activated probe, i.e. something that targets this generic activity, proteases, enzymes that cut proteins at the tumor margin, there was an opinion piece in Journal of American Medical Association commenting on this general field and on our paper. And it said one of our competitors, who has one of the more traditional approaches that targets the folate receptor, and this is a quote, while the Duke study, and Duke, David Kirsch, the clinician, is at Duke, and he was the senior author on the paper, it may have limitations. And the criticism was, well, you know, infection also has all these things going on. And, you know, to this point, my own experience with breast cancer, which occurred right after we started this, is like, you really, the tumor margin, which is actually, when you're a cancer patient, it is one of the most harrowing times. Do I have to have another surgery? Will I have, this is one of the 10 days I waited to know, were my margins clear? Mine were clear, but someone else who's now interacting a lot with the Koch, Regina Barzilay, soon got cancer after me. Her margins were not clear. She had to have another surgery. She got an infection. So this is a big deal. But the competitor said it has limitations. And you know, but are these really limitations? Because your tumor is very limited. And if I have a hangnail in my foot, it's not going to be like where we're probing. So this really was a criticism that didn't hold water for us. So just to give you a picture of this, this happens to be a colon tumor from a Roche study, but it illustrates very clearly. Here's the tumor over here, and you can see the tumor cells interspersed. The immunostaining is staining for immune cells, the brown part that you see. So this is the tumor, and you can see this enormous activity of the immune system at the boundary. And I, again, became very sensitized to how important this was when I got the pathology reports on my own tumor, and you can see this for breast cancer and others. This happened to be a very beautiful picture. And then here's the normal tissue over here, and you can see this hugely disorganized features of the tumor, and we want to, so this gives us an opportunity. All of the cells that are staining brown there will activate the probe that we have in Lumisell. So we should be able to see this very invasive margin if the surgeon has not cut it out by activation of that probe. Okay, so now what kind of drug should we use? Because the general concept of activating proteases is attractive, but how do you make it work in practice? And the devil's always in the detail. So the puzzle is we didn't want it to bind specifically to anything in the tumor. We also wanted to be able to see something. So we're going to move this device over the bed of the tumor after it's been taken out, so the residual tissue, and we want to see tumor that may be as much as 5 millimeters down within the tissue. So we need a certain kind of fluorescence that will penetrate the tissue. So that determined the kind of fluorescence we would work out. We needed a particular kind of protease activation. So we wanted something that was very non-specific. We want all kind of cells that would be there to activate it. So that's how we picked the activation scheme. And we also needed something about the kinetics. How would this drug actually enter the tumor, be activated, and get washed out? We wanted it to be commensurate with being able to inject the patient a few hours before surgery and then have great contrast at the moment of surgery so that you could see the tumor. So there were many pieces of this. We won't go through it all in detail. But part of it is the activation process. We wanted it to be activated in the tumor and not in the normal tissue nearby. And so we wanted something that would be dark when it's moving around in the blood and then suddenly become fluorescent in the tumor. And so you take advantage of properties of physical chemistry where you have a fluorescent molecule, but if you have another molecule that's similar and can absorb the fluorescence, something called a quencher, and it's shown black here because it's going to go up there and absorb everything that this guy makes. But if it's cleaved off and moves away, this will become fluorescent. So a huge part of it was to make this peptide so it would be cleaved by an enzyme, separate so it would become fluorescent. And that is a peptide sequence that's between these, no binding event, et cetera, et cetera. Okay, so these were all the pieces of the puzzle. So this just shows a molecular structure. Of course, you're going to totally memorize this, and we'll have a test later. But peptide, and there's a molecule that controls the pharmacokinetics, something called polyethylene oxide, commonly used in pharma. But the enzyme comes in. Let's see if I can make this work. And chop, chop. They diffuse weight. Of course, they're moving around, and physical chemistry isn't the greatest for physics. And then the fluorescence comes up, and it's much brighter than what is shown here on the screen, of course. And so all the puzzles came together, and we created the Lumisell 15. And there were a few more steps to it than what I'm showing you, of course. But But the success was we could show by using an antibody directed against part of the molecule, actually the PEG, who knew you could make an antibody to PEG? You can see here, and these were studies in mice, here is the tumor, the margin, the brown is the drug, the blue is the tumor core, and the red line is the invasive front. So you can see we are here at the margin, we penetrated into the tumor. Of course, we don't care about the core because this gets taken out, but you can definitely see the drug has really, really penetrated into the core. So success, and we've now had tremendous success in humans. So here is a large invasive tumor. Here's the image if you look at the tumor. Here's what the pathology shows. This is about a 2-centimeter tumor, so a little bit smaller than the tumor I had. But so you can see the fluorescence and the pathology correlate, but who cares for a giant tumor? What you really care about are these little things that are left there, because it's those that come back and kill the patient. So here, this is just 1 millimeter, this scale bar. So you can see these fluorescent images, and then when you go down and look at the correlative pathology, which is the essential thing we do, you can again see these. And this is a mark that the pathologist says, look, here's a tumor. So this is the pathology. So we've successfully got a method to image these at a very, very high resolution and find those little tiny bits of tumor that are left behind so that you can go out and take them out so that you won't have a situation like I did. Should I call my doctor and ask, was the margin clear? Because those days are really, really, really stressful. And so now Moungi will tell you about that.
As Linda said, we all have different memories, and cancer has touched all three of us, and I was also diagnosed with cancer along the way as the company was formed. And so my memory is that I was in a conference learning about cancer because I didn't know anything about cancer. And at the coffee break, David came up to me and started asking all sorts of questions that I thought were pretty straightforward questions for me. But he was unafraid of asking questions because that's his personality. And that's how he gets us to work, people to work with him and really come with solutions. And he wanted to know, can you see a single cell? And I said, yeah, single cell in fluorescence. Yeah, can you see it in vivo, something like a margin? To me, that was like, yeah, that's really easy. What's hard is the biology. You got to target the cell, et cetera. And at the time, we thought we had a drug, and so you got a drug, great, and I can make a device, a really simple device, to see that single cell, to see if there's stuff at the margin that's left over. And so that's part of the beginning. And then it was a fairly simple idea, but there was no way we were going to get an R01 fund for something as simple as what we had in mind. But it was going to be really important to build a device and to show that there was data. And that's how the Kurt and Marble Foundation, Kathy and Kurt Marble Foundation, was so helpful in getting us started, funded a graduate student part-time in my lab to build a prototype of the device, eventually funded we got a part-time postdoc, who became part of the company, and that was the beginning of it. It was really crucial to get that initial seed funding. And so going back to what was the story, and it's really about taking people with disparate backgrounds and that are not deep into the problem, to the conventional thinking, and to step a little bit outside the box. And that was really critical in getting us to where we are today. And so at the time, the conventional idea was you want to have deep penetration. You want to go find where the tumor is. And as David said, if it's a big tumor, you know that it's there. You can use x-rays, you can feel it. So it's not about finding the tumor. You don't really need the huge penetration. It's about finding, as both my previous colleagues said, it's finding that residual that's right on the surface of the margin. So you don't really need to go really deep. And then there were two points of view. One point of view was you want to have really high resolution to see the cells and determine, is this a cancer cell or not cancer cell? And another one, which is you want to go far away and see the big tumor. And so really what you want is you want to have enough resolution to be able to detect the fluorescence from a single cell, but still be able to look at the whole scene at once. basically what you want to do is astronomy. When you do astronomy, you want to look at stars. You don't see the shape of the star. You don't see the whole circle of the star. You just see the pinpoint of light twinkling in the sky, and that tells you there's a sky. And so you want a telescope that's optimized to gather as much light as possible from the star, and that's what we needed to do, right? We're not interested in seeing the shape of the cell, but gather as much light efficiently from that cell as possible. And so that also has told us what kind of resolution we wanted, but it's still looking at the big field of view, all the sky at the same time, right? The third thing was the conventional wisdom is you have this big apparatus that looks down on the patient, straight down, and big camera and everything. But that's not how surgery is done. If you look at a breast surgery or lumpectomy, there's a little incision, and then you go in and you sort of do things, it's on the side. That's not how the real world works. So we had to upend that conventional wisdom as well. And so we realized that we didn't need the big penetration. We didn't need infrared. We could go further into the visible, and that had also without going too many details, the advantage that why do people go in the infrared? It's because it penetrates farther than the visible. But at the same time, if all you care about is the surface, then because the tissue absorbs part of the visible, that also limits what you see. It also limits the signal that comes from below that could pollute what you're trying to see. And so it turned out to be actually going a little red, but not all the way into the infrared was really critical in just seeing that first few millimeters of the margin that we were after. Then we want to have enough resolution to optimize the light from the individual cells, and that meant multi-micron resolution as opposed to sub-micron resolution. And that also allowed us to have a wide field of view at the same time, so we could be very efficient at detecting this in one step. And then because the reality is that there are cavities that you have to look into, you need to have a device that can be going into the surgical cavities and look around, as opposed to have this big, massive camera system that looks down, and then you miss a lot of the tissue that's of interest. And so the device then comes with the idea that a camera is made up of pixels, and a tumor cell is about 15 micron or 20 microns in size. And the best way to optimize then gathering as much light as possible is just to have a small number of pixels, and so you create then the optics to match the pixel size or the image on the pixel size from the image on the tumor. And so that's a fairly straightforward optimization of the optics to get there. And it works. And so this is an image from a mouse model where we have single cells. This was done at Duke with our collaborator there. Single cells sort of sprinkled on the tissue, and you can see them individually here. So we knew, so we understand the optics, we understand, and then there was the biology of targeting, which Linda talked about, which was the other problem. Okay, so as I said, peritoneal, brain, breast, all these things are cavities. You have to look around, and so there's not much room. And we showed the single cell detection. You can do large area at the same time, but you're in close quarters, right? Your device has to go in there. You can't get light to go in from the side. The light has to come through the device. Right, so you have to illuminate in the cavity, and the image comes out, and everything has to be jammed together. And so we came up with the money from the Naval Fund with a prototype that Lisa Marshall, the graduate student in my lab, put together, where we put in the illumination through the same path as the image was coming out in a very simple device. From the very beginning, the idea was we want to be as simple as possible. It has to be good enough, it has to do the job, but it can't be complicated. So it's a very simple device. And then when we went to actually make a clinical device, we also wanted it to work the first time in a very simple way. And we went out and hired a third party to help us design the device. And it was really hard at the beginning because they wanted to build a Cadillac, right? That was perfect. And we said, no, we don't want a Cadillac. We just want something that works. We want it to be good enough, right? We want it to work, but not to cost $100,000, right? And so we ended up having to prove to them that we knew what we were talking about and good enough within the context of our needs was enough. And so we spent a lot of time and money then developing a modeling software to understand the optics so that we could build a device from simple lenses quickly, and it would work the first time, and it did. The other thing that was important was that working with the surgeon, Barbara Smith at MGH, understanding how the surgeon actually would be using the device to probe around a cavity. And so one of the things that was very clear is that the device had to be angled so that you could look down or you could look to the side and you could turn it around and see all parts of the cavity. And so we ended up also building a 3D model of the device, 3D inkjet printing a model of the device made out of plastic for her to test and put it around. We even put some weight in it so that she could feel the weight of the device. And we had models of tissue that she looked around, and that allowed us then to finalize the system. And that's what we built. We built it once. It worked perfectly, and that's what she ended up using in the clinical trial. Okay, then came the problem of how do you know that it's cancer? So during the surgery, it goes really fast, right? They do the incision, and they just do have to have a few. We don't want to add time. Time is money. And also for the patient, you just want to do it really fast. You don't want to change the workflow. And when you look at the image of what the drug and the device put together, it's a grayscale image. And it's really hard to see, what's brighter, what's not bright, what's bright enough to be cancer and what's not bright enough to be, and it's healthy tissue. And if the surgeon has to, think really hard, okay, is that cancer or not? That's no good. It has to be a binary decision. It has to be really fast in real time, and that's where we had to create this machine learning software. We accumulated a bunch of data, created a machine learning software to help us give us this binary decision tool that lights up then the tumor, the leftover margin, as a bright red spot, which you can immediately see on the video, and within a few seconds you know yes or no. And so that becomes now a therapeutic, right? Because now we address the cancer itself. It's a therapy. We take it out, and the patient is a clean margin. So everything is looking very good. We've as David mentioned, we've finished our phase 2, 45 patients. We did not miss any tumor compared to pathologies. We also found some residual tumors that the surgeon then in real time took out. And so we've saved a few second surgeries. We have, because of our successes, we've signed up some top cancer centers for our pivotal trial, our phase 3 trial, funded by the NCI. We've started other indications, for instance, in GI, and we hope to be available next year for patients in a commercial phase. Because of our success in the breast cancer, we've been approached by top surgeons in other indications. And so we haven't had to beat up the bush and say, we have this great idea, you want to join us? Instead, it's these surgeons that come to us and say, we've heard about your success here. We think it would be really helpful in our problems, in our field. So we're doing GI and pancreatic, we're doing ovarian, we're talking about brain mets in all these cases with our platform technology. This is an image of the device in action. There's Barbara Smith here holding the device, and there's the looking at the monitor in real time. I'm going to end with a quote from one of the patients that was in our phase 2 trial, where we ended up finding something in real time, and the surgeon actually removed a little shave, which turned out to be a margin that was not clean. And so this is what she had to say. I wanted to take the opportunity to thank you for having me in the Lumisell trial. I have had no side effects to date and doubt any will appear. The blue urine is completely gone. That's the dye that we inject. It gets cleared through the urine. I feel very privileged and grateful to have been in the trial. The results in my case could have been much different and had I not been. I wish you and the Lumisell team the very best of luck in your endeavors. It could very likely change the course of history in cancer diagnosis. How wonderful that would be. And this is really what we're about. This is what we live for, right? I mean, all three of us have gone through cancer, and we want to make a difference, and we want to save lives, and this is what it's all about. Thank you.